Don't you wish you had a clone? It'd be awesome. They would take care of the stuff you don't want to do, and whenever you need more help, just make more. Unfortunately, we can't do that quite yet, but your cells can. In fact, they're cloning themselves right now. You scrape your knee while hiking? Mitosis is involved by making you new skin cells. Too much to drink? Mitosis makes new liver cells. Tumor? Mitosis. In fact, tumors occur when cells don't die, they just keep dividing themselves.
From the time of your birth to the end of your life, this process will take place 10 quadrillion times, which is 10,000 billion times. So what happens? There are 6 specific stages of mitosis: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. For the most part, cells stay within interphase as they prepare to divide. DNA starts to replicate as it holds all the instructions for cell regeneration after cell division. In prophase, the nuclear envelope dissipates the DNA, condenses and coils up on itself. The centrosomes also move to opposite ends of the cell.
Metaphase is the longest phase of mitosis. The now organized chromatid, now called chromosomes, align themselves down the middle of a cell, while microtubes spread from the centrosomes to attach to the chromosomes. In anaphase, the centrosome began to pull on microtubes, splitting the chromosomes in half towards their respective corners. In telophase, each of the new cell structures reform, first the nuclear envelope and then the nuclei within them, and the chromosomes relax back into chromatid. Finally, a plate begins to form, known as the cleavage furrow. In cytokinesis, which means self-movement, the cell splits into two, and now you have 2 cloned cells.
But wait, if cells clone themselves, why aren't you a clone of your mother or father? The answer is meiosis, which is the separation of sex cells. Meiosis is very similar to mitosis. In fact, they have the exact same stages as mitosis, except the end product is different and we go through mitosis twice. Let me explain. In meiosis, once again, the cell goes through interphase preparing to undergo cell division. DNA replicates itself. In prophase, DNA condenses to form chromosomes, and the centrosomes move to either corner of the cell. The chromosomes stand by its duplicate copy. Here is a very important step that takes place: crossover. While standing together, the chromosomes, which are one from your mother and one from your dad, get tangled up, and some DNA is shared between the two. This is known as crossing over. This is what makes you, well, you.
In metaphase 1, all the chromosomes line up in the middle of the cell, and in anaphase 1 are pulled away from one another by the centrosome and microtubules. But a main difference is that these are whole chromosomes being pulled away, not being broken up in half like in mitosis. Then, in telophase 1, organelles regenerate similar to mitosis. Chromosomes fray back to chromatin and a cleaved furrow forms. In cytokinesis 1, the cell splits, and that is the end of run 1. The second phase is exactly the same, except here the chromosomes don't duplicate at the start. They actually get torn in half just like they would in mitosis, so it would go: interphase 2, chromatin condensed to form chromosomes. Prophase 2, centrosomes move to opposite corners of the cell. Metaphase 2, chromosomes line up in the middle of the cell and are hooked up to the centrosomes. Anaphase 2, centrosomes pull on chromosomes, breaking them. Telophase 2, organelles regenerate and cleave furrow forms. In cytokinesis 2, the cell splits, forming 4 total sex cells, all with 23 chromosomes waiting to bind to another sex cell, either sperm or egg, to make you and all of your features you see today.